So what questions do you folks have? Yes?
Can you post Friday's recording?
I have not posted Friday's recording. Okay, well, it's a good thing. I think I didn't just delete it. As long as I didn't delete it, I'll do that. I'm sorry. My machine does not have much storage on it, so I have to be really pretty efficient about deleting these things. But I don't think I just deleted it because I wasn't sure whether I'd posted it or not. So I will. Yes.
Can we talk through the serine protease mechanism again?
Yeah, absolutely. Let's do that. So serine protease mechanism. So that would have been what we did last, right?
Mm-hmm.
We did it right here. Let's go ahead and just grab the picture. And this thing seems like a giant memorization fest, and to some extent it sort of is. Were you okay looking at it step by step big, or do you want me to zoom out real quick first?
Step by step.
Okay, we'll just do step by step big. Can everybody see this? It's a little bit blurry based on the figure, but so once again, you need to know this mechanism, and the way I will test it, if you look back at the old exams, there's got to be at least one example of this, if not both exams, where I'll pick some step in here and I'll say, okay, give me the next step. And what I'm asking you to do, if that's this, if it was the first step I was asking you for, which I could do, I can do any step, but if it was this first step, what I'd be looking for there is for you to draw the arrows to show me where these things are going. And once again, this is happening in an enzyme. So in biochemistry, we often draw these as concerted, all of them happening at the same time. And I haven't read the primary literature enough to know whether that's really what goes on or that's just how we draw all these things in biochemistry. I think if they were very not concerted, it would happen that way. Because we definitely see things where we pop electrons up and then pop them back down, right? But anyway, what I'm looking for here is draw the arrows and then draw the products down here. All right, and so a couple of things here. First off, this is for chymotrips and the numbers on these residues. I don't care about those numbers, okay? They're going to be different in any serine protease. They're going to have different numbers on those residues. Yes?
Since you'll give us another step, we don't have to memorize those molecules exactly, right?
You don't have to memorize these things. The only thing I have done before is I have gone in and said, okay, draw me the next step, and you might need to draw another molecule. I don't give the water. I don't show the water. So I have done that once, but so that's a possibility. But otherwise, you don't need to have the structures of the amino acids outside of needing to have no structures of amino acids in general for this test. And I think I asked you to draw the peptide for the last test, yes? Yes. So this one you would just need to, maybe if I asked for a beta sheet, you'd need to know 6 or 8 amino acid side chains of your choice. Yes, Claudia.
The bond between nitrogen and carbon, there's highlighted in red. Is that for a specific reason that should be paid more attention to that?
What are we looking at here?
So
Here? Down here, this is the one that breaks, or right here, this is the one that's going to break, is what they're talking about. So that's why that's highlighted in red there. What was that?
Just not in that step yet?
Just not in that step.
Okay.
So we'll see it. It's actually breaking right here. We haven't started here. I'm going to keep answering questions. Yes?
In the next little thing, it has the, are those dotted lines with the plus between the two nitrogens? And
that's good. And that's something I wanted to ask about here. Oh, here.
Yes.
I wouldn't worry about that.
Where did that come from?
That's resonance.
Oh, it's just showing the resonance.
It's resonance. If I have this protonated and that protonated, we're then sharing the double bond between these two. Okay. So it's 1.5 bonds. Yes.
Is that also what they're trying to show with the 2 oxygens and the carbon?
Here, yes. So this is whenever you have a carbonyl, you shouldn't ever think about this as actually being different. So if I have a deprotonated carbonyl, let me get this down where I can draw.
For the purpose of today's exam, are we going to draw it like that when we're showing the resonance?
You can draw it either way. Okay. But this, we have a resonance form between these two, right, that does this, which is why these are so stable. But you could also just draw it like this, and that's actually more correct. Okay. So So you asked about these dotted lines, and you could draw those dotted lines if you want, but I wouldn't worry about them one way or the other. But these dotted lines right here are hydrogen bonds, and I don't really ever take up points if somebody draws them, but I don't care about those either. I don't care where the hydrogen bonds are. If that makes sense. So this picture has them, but I'd just as soon you not worry about them. Let's see. Let's do a nice thick white. And so if this wasn't here, if that was gone, and this was gone, you know, that would be just fine. In fact, that would be my preference, but I won't take off points one way or the other. Does that make sense? Okay. So, so let's go. You guys, any other questions on details here? And shall we go through the mechanism? All right. So we start out. This is our catalytic triad, right? You should know the rules of the things in the catalytic triad. So what is the serene's job in this mechanism? What's the serine going to do if we think about the whole mechanism?
It makes the covalent bond oxidation.
Right. It's a nucleophile, and so it's going to attack the substrate, and we're going to get a covalent bond there. So this is a part of our covalent catalysis, okay? But you know, the big thing is it's acting as a nucleophile for us. What's the role of the histidine? It's to deprotonate the serine in this step, right? But it's actually going to act as an acid and base. So through our mechanism, it's going to take a proton twice, and it's going to give back a proton twice as we go through. Once it's interacting with serine, and once it's interacting with water. Actually, it's not with serine the whole way for this, but it does take a proton twice, and it gives off a proton twice. What's the aspartate doing? Why is that needed?
Changes the pK of histidine.
Yeah, it changes the pK of the histidine. So having a negative charge here makes this histidine more stable and being positive, right? So it raises the pK, it makes the histidine better at taking protons. And this is a pretty hard proton to take. The pK of this is 15, right? Now there's more in free solution. There's more things going on here, but not an easy proton to take, and that's why this is such a great nucleophile once you take it. All right, so first step here. This histidine is going to pick off this hydrogen, so the serine can act as a nucleophile and attack this carbonyl. You kick the electrons up to the oxygen.
Can we write those down?
Can we write what down?
We talked about the covalent cat. We talked about the three parts and what they did.
Yes.
Can we write that down?
Oh, for right now?
Yeah.
Okay. You also could go back to one more day. If I don't see them fully written, here we go. How about that? So these are the notes from Wednesday, last Wednesday, October 22nd. Can we just do that?
Yeah.
Okay. Or actually, I mean, I don't need to be that stingy. Because I have technology and I can just do this. Bonk. All right. Just copy this over. Okay. All right. And we all want to remind you, we refer to these as the catalytic triad. Okay. So going back, we just did that first step. We kicked these electrons up onto the oxygen, and in organic chemistry, whenever you kick electrons onto an oxygen, what happens? They come back down. And when they come back down, what happens? You kick out some leaving group, right? If it came down and they just kicked out what arrived in the first place, nothing overall happened. Doesn't mean it doesn't happen actually in solution, but for us to actually have the reaction go forward, we have to kick out something, kick out a leaving group. So these electrons, that's a pair of electrons there, come down, they make a double bond. These electrons here, if this bond gets broken here, these electrons go up and pick this hydrogen off of this is the hydrogen from the serine. They pick them off of the histidine, and then this is going to leave. So we just broke our bond, and the half that peptide is leaving. And that was the whole point, right? That's the whole point of this mechanism. And then the rest of this is going to be getting rid of, let's see if I can erase these hydrogen bonds. There we go. Okay, and the rest of this mechanism here, so these are picking up this hydrogen, these electrons go here, this leaves. The rest of this mechanism is about regenerating the enzyme, essentially, right? I broke the peptide in half now, that's the goal of this enzyme, but we have this piece of the enzyme, this piece of the peptide that was being cut, still attached to our serine protease, and we can't cut another peptide if we still have this attached. So that's what you see down here. Okay? So now the rest of this is going to be, we need to break this bond and put a hydrogen on there. And we're going to do that using water. So what happens here is that water comes in. The histidine can pick the proton off the water, leaving us with a hydroxide, which is a great nucleophile. It attacks this carbonyl. We kick the electrons back up again. Whenever the electrons get kicked up on top of the oxygen in organic chemistry, they come back down and they kick out a leaving group. In this case, the leaving group is the enzyme, right? This is all attached. This is all big molecule and enzyme. And this serine, that is the leaving group, it picks off this hydrogen as this leaves, and these electrons go back up onto the histidine. Our other half of our peptide leaves, and we have regenerated our catalytic triad in the enzyme. Okay, so when you're thinking about this as well, think about how we've got two steps, right? We've got two steps where we kick those electrons up on that carbonyl. There's two steps where you kick them up, they come back down, and they knock something out. And you might look at it, you might look at what step you're in and see what needs to leave, right? If we're going to kick up those electrons and we're right here at the beginning before we broke this peptide bond, well, what's going to need to leave is this half of the peptide. And if we are halfway through, or we've got our covalent intermediate here, we kick those electrons up, and then what needs to leave is the rest of the peptide, or actually from the perspective of this carbonyl, the enzyme leaves. Yes, Robin.
Why does the hydrogen now have a dotted line? It's in the middle of the.
This one right here?
No, on the other side. You see how it has a solid line between the hydrogen and the nitrogen?
Right here?
You go to the next step and it's dotted.
Why is it dotted?
Yes.
You know, they like to, this book likes to draw that as kind of a between the two. You know, and maybe that's more of how it exists when you delocalize all those electrons, but don't get obsessed with the delocalization of electrons or these hydrogen bonds. Right. Well, hold on. Was that a bad one to do? No, that one was okay. You know, and I would get rid of these just to make it cleaner. And with the questions you guys have had, you know, for years I've been using the one out of your book. Maybe I should just find a different mechanism that doesn't have all the hydrogen bonds drawn in. A little bit confusing. Okay. There is not one set. Let me look this over here real quick. But there's not a set of dotted lines on here I care about. Okay, it's a dotted bond, it's a hydrogen bond, it's delocalized electrons. As long as you recognize these are delocalized electrons and draw it the correct non-delocalized form, I'm okay with that. And if every place you've got hydrogen bonds, if you don't draw them, I'm okay with that. You of course you can't have the hydrogen floating out in space. You'll have to covalently bond it to something. Questions on that? Yeah, Bridget.
How is it like all together if the piece that the aspartate is on is only hydrogen bonded to ONH?
How is all this connected?
Well, so like there's an O dotted line H to the other pieces while, like, yeah.
Nope.
So it just, but how, okay.
It's just a hydrogen bond. I wouldn't worry about it.
Okay.
So the aspartate is here. I would view this as to provide a negative charge next to this, next to histidine, to help the histidine grab a proton and become positive. It makes the positive form of histidine more stable, which makes it better at pulling that proton away from that serine. Yes? Where would I put this negative charge? I would put it there, actually. I mean, right, you've got these delocalized electrons, but over here you've got a positive charge. They're going to move this way. They're not going to be, it's going to, everything reacts with everything else. So, any other questions about the mechanism? So just do this like a standard, do these like standard organic mechanisms, okay?
We're just gonna have one step.
So just have one step and do the next step, and I won't give you any, and your step will not have these, whatever I find will not have these hydrogen bonds. I'll either erase them or whatnot, okay? Other questions? What else would you guys like to ask about?
Can we go through more examples from what we were learning at the end of class on Friday?
I'm gonna have to remind myself what that was. Oh, changing pKs. Great. Yeah, that's important. So let's think about, clearly went to the wrong place. So let's think about what happens when, so like I set up above, all of these guys, all of these amino acids react to the presence of other amino acids. So if we think about, like right here, this histidine, its pK is going to go up, so it's more likely to be protonated, right? And this aspartate, what's going to happen to its pK? It's also, well, it's actually going to go down, so it's more likely to be deprotonated. Okay, each of them affects the other. All right, so that's not my only example, but so let's go ahead and say, let's look at that. So let's say we've got, and well, I guess this is a glutamate the way I've drawn it, right? So my glutamate is in the active site of an enzyme, and right next to it, that's not, hold on. Let's say right next to it is another glutamate. And I just drew them like that, all right? What is going to happen to the pKs? Or let's say, how did the pKs of these glutamates change? All right, what do you think? So let's go ahead and just really quickly draw up here. So glutamate's usually got a pK of 4-ish, okay, on that. And so above, below the pK, what form is going to be present? Neutral. And what's going to be present above that? Negative. Okay, it's going to be negative up there. So which of those forms is going to be favored if I've got two glutamates next to each other? The neutral form, right? Because if I end up with two negative glutamates next to each other, that's going to be repelling. They're going to repel each other, and that costs energy, right? You have to put energy to have that happen. It's less stable. All right, you can't see either one as glutamate right now. Neither one is negative, but so they're each going to affect each other there. unless you had something else going on. Neither wants to be negative in this case, and so it'll go up so that we're less likely to end up with both negative. Yes.
So both of the PKs go up.
Yes. Without any other information, that's what you would assume. Now, if I was to put a positive charge over here, but this is more complicated than what I would give you, but if I give a positive charge over here, then this one's more likely to be negative, right? So that would, putting a positive here would force its pK down as opposed to up. But then this one isn't going to want to be negative. Did I say it wrong? No. The pK needs to go up. So the range at which they can be neutral is larger. Right? If I raise the pK, there are more conditions under which it's neutral.
Oh, my God.
Yes.
So why doesn't is it kind of like circular almost? Like one goes up, so then the other one goes up instead of being negative?
It's a little bit circular, I agree, with this sort of thing. We can't tell which one's going to be effective, right?
But if you drew it as one was negative and one was neutral, or the negative one would make the neutral one.
If I had one of these deprotonated, so it's even more obvious that way. If I have one of these deprotonated, right, with the way I draw it, I say what happens to the pK of that? Same thing, right? For this one, that pK is going to go up because you don't want to have a negative next to a negative.
Okay.
So that would be a more obvious example. What if I have, say, a lysine? And I'm going to draw this lysine in its positive form, since we're going to talk about pKs here. And next to that lysine, I have an aspartate. Okay. What happens to this system? What happens to that pKa if you have this situation? So what happens to the pK of lysine when it's next to an aspartate? And the pK of lysine, let's say, is approximately 10. One of these days I'll look it up and memorize it, but somewhere in there.
Would it just not change?
Okay, so let's look at this. pK of 10 in free solution, right? When I talk about that, it has a pK of 10. It's what its pK is when it's by itself with nothing else around but water. All right. So up here above, what's the charge state going to be above the pK? Neutral. And what's the pK going to be? What's the charge state going to be below the pK? It's going to be positive. So we just made the positive more stable or less stable? More stable. So you want to have the positive around under a greater set of conditions. So as it moves up, you widen the range for the positive. First, Sophia?
Okay, I think I'm just misunderstanding. So I hear like the low is positive, the other one below. How do you know?
How do you know whether I put the positive here or a neutral there? Yeah, that's good. Good question. So when we think about this, the way I like to think about it is, so whenever you're below a pKa, you're going to stick protons on. Okay, you're below. So think about your pH relative to pK. When pH is lower, you have more protons around, and they stick to things.
Okay.
And when pH is higher than the pK, you have fewer protons around, and they come off of things. That work?
Yeah.
Okay, so then when we think about our two possible states here, I'm just drawing a positive, but we have NH3 plus and we have NH2. Okay, and then over here we also have our possible states are protonated carbonyl, or I'm sorry, carboxylic acid, or a negatively charged one. Right? You're welcome. Yes.
Why aren't both forms like favored? Because like in the previous example, if there's a negative, then the other one's neutral. So why is it because the neutral was kind of like it's like the better form of the options, you want the better form of what's available?
Which one are we talking about here?
Like the one above.
Okay.
Like one state, it favors the neutral.
Mm-hmm.
In this one, it favors not both the neutral and the positive, it just favors the positive. Is it because
It favors
it's possible to be positive?
It favors the positive here, and this negative here would also be favored.
Mhm.
They would occur both. They would both influence each other. So if I asked you about this one instead,
Mhm.
you would say the negative's favored. So you would have the pK of the carboxylic acid, the aspartate, would go down to make it negative all the time. Does that address your question?
I think so.
Okay. Yes.
What if instead of the negatively charged aspartate, you had another positively charged lysine?
That's a good question.
Would one of them favor the neutral state, and then one also favor the neutral state?
Yeah, let's think about that. So let's go ahead and put one lysine there, another lysine right here. That's too many carbons. Hold on, let me fix it. Okay, so I've got two lysines here. And so what happens to the pKa of either lysine? I'll say each lysine. Right, so we can scoot up here to look at our thing. So we want to, with two positives next to each other is less stable, right? You have to put in energy to have two positive charges next to each other. And so proton, I want to dissociate from one of those. So I think you said lower. This gets lower, so we're more likely to have one of them be neutral, or both. What is moving the pK?
Yeah, I don't understand, like, if they want to be more, if they want to be, like, higher.
They want to be neutral.
Yeah, they want to be, yeah, so what is the PK?
The charges, the fact that it's less stable.
Okay.
So the fact that it's less stable to be like this means that the pKs move so that under a greater set of conditions, they're neutral.
Okay. And then below that would be positive, right?
And below that, below whatever the new pK is, it would still be positive.
Yeah. Okay.
If that's what at the pH value, that's below the pK, right?
Right.
So when we use pKs, we're always thinking about what pHs are there. But one of my professors in graduate school, right off the bat, somebody said, "What's the pK that?" And she said, "Well, you don't know what the pK is in a protein, right? I mean, you do, and part of the research I do involves tools that you're trying to predict what the pK is of a amino acid side chain in a protein. But pretty much none of them are the standard pKs. Okay, if it's on the outside of the protein and nothing's interacting with it, maybe. But otherwise, there are other things around it that influence it. Is that good enough? Do you guys want to try another? Actually, do you want to try let's try a hydrophobic example. So let's say I've got let's say I've got an aspartate. We'll do it in its protonated form, and it's in the inside of something. There's some pocket here, and for some reason that pocket is just lined up with valines everywhere. All the valines you can stand. Okay. What's going to happen to the pKa of my aspartates? Remember, that pKa is about 4. And then down here we are in this form. And up here, above the pKa, we're going to deprotonate. What happens to that pK in this situation? It's going to go up, right? So the pK is going to go up. So this neutral form is around more of the time because charges don't like to be, I mean, hydrogen bonders don't like to be next to hydrophobic residues, but charges are even worse in terms of energy and stability.
Can you explain it one more time?
What was that?
Can you explain it one more time?
Right. So when you think about it like an extraction in organic chemistry, right? When you wanted something to go in the water phase, what did you do in organic chemistry? You made it charged, right? You protonated it or deprotonated it, whatever was the way to do it. You make it charged so it really likes to be in water, as opposed to only sort of likes to be in water. Okay? And this isn't in water. So you don't want to make it so it really wants to be in water, and then you stick valines all around it. Did that help?
Yeah, so you're avoiding making it charged because it's hydrophobic.
Right, you're going to make it more hydrophilic. Right now it's already hydrophilic, but you're going to make it more hydrophilic by making it charged. All right, any other questions on that? Right. Questions on anything else? Yes, Robin.
How do you calculate the dissociation constant with no equation and only a graph?
Okay, good question. So you're looking at an old test there, right? So we've got some graph there, right? Did it give you? Does it have the graph?
Yes. Would you like it?
I'll make up one, and you can tell me whether. So let's say I've got a graph for, it's probably antibody binding or something like that. I've got my ligand concentration down here, right? And over on this side, I've got my Y, which is my fraction bound. Or it could be a percent, right? But fraction and percent, you should be comfortable with that. So this could be 1 to 0. Was it a fraction or was it a percent in there?
Oh, fraction.
Okay. But otherwise, that'd be 100%. And then, so a curve where you actually got it all of your ligand to bind, right? Equilibrium might not be all of it bound, might look like that. And if you remember, so if we look at our equation for ligand binding is this. Okay, our equation for ligand binding looks like that. And we did the algebra for this, but when half of our sites are bound, when half of our protein sites are bound, our KD is equal to our ligand concentration. And you can see that by, if let's say 4. If I put 4 here and 4 there and 4 there, I end up with 4 over 8, right? You guys with me for that? So 4 over 8 is 1/2, right? Anything I put in here where this is equal to this is going to give me 1/2 out. So the math says that it's where half of these things are bound, that's my ligand concentration. So let's say on the bottom here, I had, this is, I don't know, 1 nanomolar. This is 2 nanomolar. Then what I could do is I could read at 50%, I read over, I hit my graph, I read down, and that to me looks like maybe it's 0.33 nanomolar. 0.25 maybe. Let's make it 0.3. Something like that. So it's where half of my sites are bound, that's the concentration of ligand that's equal to my KD. So in other words, KD is this. Does that make sense? Okay. And it comes out of the math. Yeah.
So when y, oh no, y is equal to 0.5 or like half is when you go down. You
Right.
Okay.
So what if this didn't get? What if the graph was drawn, but it looked like this? Actually, let's not do that. Let's make it a little bit taller. Okay, so it didn't quite get to 1 there. Where would I read this? Nope, I'd still read it at half because it's half the sites. So this might represent an equilibrium, like at equilibrium, all the protein sites aren't bound, which can happen. And so I'd still be reading at 1/2. I'd still read at 1/2 all the way over. But that's going to maybe have a 0.5 nanomolar KD. Does that make sense? It is actually literally, the math says, it's this equation, and the math says it is literally 1/2, not 1/2 of how much you were able to get bound. Yes?
So we're always going to be reading at 50%.
You're going to always be reading at 50% for this. Now, if you were in lab doing this and you got a curve like this, right? And you were doing it versus some indirect measure, you might have to assume that that's 100% and hope for the best. Okay, other questions or questions about that? About ligand binding?
So with half of the ligand bound, the KD is equal to the ligand concentration.
Correct. When half of the protein sites are bound, when half of the protein sites, not the ligand. You've got lots. One of the assumptions here is you have lots more ligand than you have protein sites. Okay. So when half of those protein sites are bound, then your KD is equal to your ligand concentration. All right, good question. Other questions? Yeah, Nick. The R state and the T state and that sort of thing? Yeah, okay. Let's go ahead and look at that. I'm going to maybe go back over the old notes here. Otherwise, I'll spend 5 minutes pulling pictures across. So hemoglobin and hemoglobin has this R state and this T state, okay? And what's going on here is the R state and the T state. So we've got quaternary structure here, right? We've got 4 subunits all associated non-covalently, and they change shape. All right, so we have the T state where it's kind of more open in the middle, and we have the R state where things kind of close down. But for our explanations of how these substances affect which state we're in, where these substances are, we're talking about carbon dioxide or protons, right? Or 2,3-bisphosphoglycerate or O2, right? And carbon monoxide is different. Right? Carbon monoxide and carbon dioxide are different. They bind to different places. But how these things affect this depends on, you look at the differences in the states, and for our very particular examples for carbon dioxide and for protons, note that we have groups up here that are close together, right? This right here is close to these groups in the T state, and it's way down here as opposed to there and there in the R state, right? So we can have stabilizing for those specific examples we talked about. We have stabilizing interactions in the T state, but not in the R state because of distance. All right, so this is our basic. We'll come back to oxygen. It's more hand wavy. All right, so this is an example of how, let's see, protonate. So here we're talking about protons, all right? So for here, how does the acid concentration, so in your tissues, pH is about 7.2, in your lungs, it's about 7.6, and how can that affect which state we're in? Well, we've got a histidine here that's on the N-terminal end. That's not as important, but we've got a histidine here in the R state. I'm sorry, we've got histidine here that when it protonates in the T state, it is right next to an aspartate. So we get this great positive-negative interaction. So when you bring a positive next to a negative, you release energy that makes you more stable. If energy is released, things become more stable. So the T state, they're next to each other. But in the R state, I don't have a close-up of the R state. In the R state, here's that histidine and here's that aspartate. They're far apart. Here they are in the T state. Okay, so that stabilizes the T state because energy is released when you're here and energy is not released when you're here. And in fact, going from here to there, you have to put in regards to this interaction, you have to put in energy to go from there to there, right? Just like bringing charges or opposite charges closer together releases energy, pulling them apart takes energy. Okay. More generally, this sort of thing is happening all over the hemoglobin. Okay, so all over the hemoglobin molecule, you're taking places and you're making positive interactions, okay, when you are in the T state, and you're maybe breaking positive interactions from the R state by protonating, right? You make some things neutral, you make other things more positive. Does that make sense for that one? But the most specific example is this histidine interacting with that aspartate. For carbon dioxide, which was on your quiz, carbon dioxide is going to attach to the N-terminal end of each subunit. So this is how hemoglobin carries carbon dioxide. It does not carry it on the heme. It actually makes this carbamate, which has a negative charge. Now we have this negative charge on that N-terminal end of each subunit. And at least for this subunit, that N-terminal end, it's actually right with that histidine, that N-terminal end that is now negative is next to a lysine on this other subunit. You see that? So lysine is positively charged, so I now have a negative and positive charge next to each other, releases energy, makes it more stable. If hemoglobin wants to switch to the R state, these two things are not near each other. Okay, so we don't get that stabilizing interaction. And in fact, if you were here trying to go here, you'd have to actually put in energy to break that interaction. Does that make sense? So for carbon dioxide, all we have is the specific example because there are 4 carbon dioxides that bind. In terms of if we talk about protonation, there are more generally, more sites that protons can bind. So then, if we want to go and talk about oxygen, this is a more hand-wavy explanation in terms of I'm not going to be able to talk, it's complicated, I'm not going to be able to talk about all the, or talk about it exactly in as much molecular detail. So here in the T state, this iron is pulled back, right? It's back, this helix is back, and this helix is part of the whole rest of the subunit, right? I'm not sure where this helix actually is in, you have a picture here. I'm not sure where to identify that helix exactly in the picture. It's maybe this one for this heme. But this is puckered back. When oxygen binds to this, what happens to that is that oxygen, it pulls that iron forward. The histidine's attached back here to the iron, so this histidine pulls forward and pulls this helix forward. And all those helices are in the hemoglobin together, right? That subunit. The subunit changes shape as a result, which causes your other 3 subunits to also change shape into the R state shape and be more likely to bind oxygen. So that's how we get oxygen helps more oxygen bind. All right, so but it's more of a hand-wavy global things moving around on the protein sort of thing. Yes?
So if on the exam you asked how oxygen, like how it favored one state or the other, and we said like it changes the shape by like pulling it forward, would that be?
I would need more details. So
Okay.
the oxygen binds to the iron, which pulls forward in the heme and which pulls the helix behind the heme to pull forward.
Okay.
which causes these changes. Then you could go into the more general shape changes, right? But the specific parts of this is oxygen is binding the iron, it is pulling that iron forward. You can talk about being puckered and flat if you want, which pulls the helix behind the heme forward and causes global changes. Other questions? Yes, Lauren.
Go over like the phi and psi.
The phi and psi angles. Yes. Is that good enough? Does anybody need to see 2,3-bisphosphoglycerate? All right, let's talk about phi and psi angles. And I'm not sure. To get back until we see a Ramachandran plot here. All right, here we go. Phi and psi angles. So when we think about orientations of peptides, so here's a peptide, right? Here are peptide bonds. So these peptide bonds, essentially, we have a resonance structure where we could take the electrons, we can kick the electrons up here. Did I draw that? Yeah, there we go. So what we've got is we have a resonance structure where these electrons can come down, we can kick them up onto the oxygen, and so we have a double bond here, right? So what that really tells us is we could draw those dotted lines. This is more like 1.5 bonds, and that's more like 1.5 bonds. Okay, probably not quite that, but well, let's treat them like that. And so we can't rotate around this bond. We don't rotate around this bond. This is all one plane here. Okay, and so these two angles right here, if we think about a specific amino acid, right, go carbonyl to, there'd be amine over here, these two bonds are the only ones that can rotate in a peptide. Right, so those are the two we need to concern ourselves, and those will determine what the global shape of that peptide is. Because the backbone is going to determine, where the backbone goes is going to determine your shape. The side chains can kind of flop around, right? And the side chains can interact with each other to pull the backbone around. But really, if I know what all the angles are in the backbone are, I know what the global shape of that is. All right, so one way to represent that is what's called the Ramachandran plot. And what this is this is a plot of each phi-psi angle for each amino acid. All right, so I got my phi angles, my psi angles, and so let's pick off a dot. We've got one right here. Okay, let's take that one. So if I take this dot right here, that whatever amino acid that is, that has a phi angle of what looks like about 40, let's say. Let's say that's 40 degrees. Okay, positive 40 degrees. And it has a psi angle of about, looks like about 60 degrees. I don't know. I'm not going to make you, I mean, it's going to be, well, they're both next to a double bond. It just depends on which, the first one or the second one. So if we think about, it is phi psi in order. So actually, I do probably know, but that just accidentally. So phi is here, psi is here, but they're both next to a double bond. It's whether the double bond is next or whether it's before it. I don't expect you to know which one's phi and which one's psi, okay? But so for this amino acid right here, this guy, I'll go ahead and do it in, oh, you were probably looking at that. I'll do it in orange. This is going to be our phi, and that's going to be 40 degrees. Positive 40 degrees, and that's going to be our psi for this example, and that's going to be plus 60 degrees. C. Sorry. Brain failure there. So 40 degrees and 60 degrees. That makes sense kind of what's going on there. And so how would I ask this? If I wanted to ask if you understood this, I'd probably actually do the reverse. I'd probably say, here, let's just add even more confusing color to this. I'd say this one and this one, let's say this one is negative 50 degrees and this one is negative 120 degrees. Okay, plot that. All right, plot that. And so what you would do is you'd say, okay, and I'd tell you which is phi and which one's psi, because I just said I want you to memorize that. So I go minus 50 is going to be about here, right? Okay, and minus 120 is about here. So it's out here, and it's some weird, isn't a real regular structure. So it's something really weird.
Would you ask us if it was right-handed or left-handed?
I could ask you what secondary structure it is, but I would have those labeled if that was the case. This figure doesn't have them labeled. Well, it does actually, but not officially, right? So if I given you something, I could have like been more careful with the angles and made this 120 plus 120, like that. In which case, this dot would have been up here. And then you would have to tell me it was a beta. I could have asked him what secondary structures, and you could have told me it was a beta sheet. All right. So mostly I'm going to see that you understand this plot enough that you can plot something on it and read something off of it. And this is something that, protein chemists use a lot. If I plotted something and I ended up with an angle here, something's weird, right? So maybe it's something weird and really interesting, or maybe it's weird and there was an error. Yes?
Just to read that, is all of the data sheets, or is that?
This is beta sheet up here. This is alpha helix right here.
Okay.
All right. You guys have a good couple of, well, I'll see you tomorrow.